Welcome. My name is Suzanne Fetherling. I'm super excited to have you here. I'm just like you. I'm currently a high school teacher teaching a bunch of different levels of biology. The lab I'm going to introduce to you today is our Photosynthesis Cellular Respiration lab that's put out by BioRad. Everybody in the program is going to be doing this particular lab, so hopefully we come up with a lot of great ideas on how we can use this with our students. I think that this is a great lab because it's a lab that you can really ramp up for your older kids or higher level students, and then we can also present it in a much more simplistic manner if we're teaching younger kids or lower level classes. It can be used in many different ways. It's also really great for any elective classes that might incorporate ecology or cellular energy, even some chemistry classes as well. You should have received a box that looks like this. It is BioRad's Photosynthesis Cellular Respiration Lab. There's a bunch of things inside this box that are going to be very numerous that we're probably not going to use everything because we don't have a full class set of kids, but you will be using all of the materials. One of the first things you'll notice is you're going to get this white styrofoam box. It has 100 cuvettes in it. I will show you how to use these a little bit later. This is definitely plenty for a class set, even if the kids are going to set up multiple samples. Definitely hang on to those. They say disposable cuvettes, but I always save mine. I have a ton of cuvettes at school just because I've gotten numerous boxes. You're also going to get a bag of caps, which these caps really do keep the cuvettes sealed pretty tight. So we're going to end up adding some liquid volume to the cuvettes, and these caps allow us to kind of lay the cuvettes on their side and keep them sealed. You will also see that we have pH strips where the kids can use this guide here to measure their samples and see how changes in pH occurs depending on the levels of photosynthesis or cellular respiration and how they balance each other out. So I'll show you how that works as well. You're going to get some transfer pipettes. This is pretty much just to move liquid around and do some very simple basic measurements. We can use these. One other very important thing that you're going to see is there's going to be a temperature sensitive package. You're going to want to throw this in the fridge. It is not, if it gets left out for 30 minutes or so, or even longer than that at room temperature, it's not really going to hurt anything. I would just try keep it out of the heat. I would put it in the fridge as soon as you can. There's going to be 3 bottles in there. One is going to be a CO2 indicator, which I'm going to show you how to use in a second. The next is going to be our very beloved and very cute algae beads, which we're also going to look at in a second. And the last thing in here is something called de-beading solution. The de-beading solution I'm not really going to use over the next couple weeks. You can try it if you want, but this is just used for taking some of the algae beads. You're going to de-bead them or release the algae out of the sodium alginate, which I'll talk about later. The kids can then look at their algae sample under a microscope. The thing about this lab is a lot of people are a little bit intimidated by setting it up. Really, it's a very easy lab to set up. You just need to do a little bit of executive planning. The 2 first things that you need to do before you even get stuff ready for a class, if you were doing this in school, you have to make your CO2 indicator solution, and then you also need to activate your algae beads. Those are the first 2 things that you want to do, and once you get them done, they can sit and they're good for a while. So you can do this whenever you want and then have things ready to go, so once you're in school, then your supplies are ready for whenever you'd actually like to start the lab. So to make your CO2 indicator solution on this bottle, this is a 10x concentration CO2 indicator solution. So the only thing that we have to do is dilute it. If it says 10x on the bottle, that means it's 10 times as concentrated as what we want it to be. I already made myself some CO2 indicator solution, but all I did here was I took 10 milliliters of this 10x concentration CO2 indicator solution, and I added it to 90 milliliters of spring water. And my spring water, I suggest that maybe you get this. So this is week 1. This would maybe be a good thing to mention. I went to Jewel just right across the street. I got myself a gallon of spring water, and I'm gonna use it in a bunch of labs over the course of this month. To make this CO2 indicator solution, I used 10 milliliters of this 10x concentrate and 90 milliliters of spring water. Then I just put aluminum foil on it, not necessarily tight, just kind of covered it, and I set it off to the side. This CO2 indicator solution needs to sit for at least 24 hours before you use it. So once that's done, just keep it at room temperature. I left it out on this little table that I'm working at right here. And that's all you have to do for the CO2 indicator solution. The next thing that we're going to do are the algae beads. Before you activate the algae bead, you have to have this CO2 indicator solution ready. So that's why I already pre-made this because this has been already sitting out for about 24 hours. So now I want to activate my algae beads. And when we do this, they are in a storage solution. All I'm going to do is take this and I'm going to actually dump all of the algae beads into a little beaker. So I've got my algae beads and then I've got the storage solution that's on top of it. These transfer pipettes come in really handy. There's a little flap. You can open them on the bulb end of the pipette, and then we can kind of just scrape out the rest of the algae beads that are still in this light sensitive bottle, which I think I did a pretty good job actually of getting them out. I'm going to squirt a little more solution in there, try to get as many out as I can. To activate our algae beads, the first thing is we want to get them out of this storage solution. All I'm going to do is decant the storage solution and pour a lot of that off. Just take this transfer pipette and pipette off some of this excess storage solution. Now, if you look in the BioRad manual, they kind of have all of these real specifics on how you need to do this. Make it easy on yourself. All I'm going to do now is I'm going to take some spring water. I'll pour some in a little cup here. I'm going to take some spring water. I'm going to rinse twice. So literally, I'm just going to pour some water over my beads. I'm going to pour this water off. Pipette off what's ever left over. And after I've rinsed the beads twice, then I'm going to let them sit in the spring water for an additional 5 minutes. So again, just taking some of this water off. So I've rinsed them twice now. I think in the manual it says like 25 milliliters of water. I've never measured. We just want them to sit in some spring water at the moment, because then if any of that storage solution has penetrated these algae beads, we have time to kind of get that out. If you let them sit for a little bit longer, it's not a big deal. So you can see that this CO2 indicator solution is a nice amber color. And if we look at our indicator chart, this is pretty much right in the middle. So it's a very neutral pH. And the reason that we had this sit out for 24 hours, 24 to 48 hours, is because we wanted it to equilibrate with the atmospheric CO2 and get to a nice neutral spot. Now, our algae beads, what we're gonna see is that when you activate them, we're gonna put them under light immediately, and they're gonna start photosynthesizing. And when that happens, they're gonna absorb some of the CO2 that is in this indicator solution to use for photosynthesis. When that happens, then this indicator is going to become much more basic. And I think I have this upside down, but I'll be consistent here. Sorry. So it's going to become more consistent because that CO2 is being consumed, and that means we're reducing the level of carbonic acid in our samples. We're going to turn more purple and become more basic. When the beads have been under no light for an extended period of time, the process of cellular respiration is going to take over. They're going to be releasing CO2 into this indicator solution, which turns into carbonic acid, making this more acidic, and our samples are going to start to turn yellow. These have now been sitting for about 5 minutes. I'm going to take this water off, and when I do that, we're going to add the CO2 indicator solution. Oops, I think I lost a bead there. So pipetting some of this water off, and then adding our CO2 indicator. And I'm not going to use any particular volume, just to make sure that there's enough coverage for them and that I have enough CO2 indicator solution left over for actually testing my samples, which you can always make more. You just want to make sure that it sits out for at least 24 to 48 hours. All right, welcome back. So I'm ready to distribute my beads through my cuvettes depending on how many samples that I want to test. So just a reminder, things that I've done with the beads already. We took them out of the light sensitive vial that it came in the package, rinsed them with water twice, let them sit with water for about 5 minutes, and then put CO2 indicator solution that was already diluted on top of the bead. And I let those sit at room temperature, just an ambient light, for about 6 hours. And then I threw this beaker under the light source that I had for about 20 hours. And then I removed this beaker from the light source and threw some aluminum foil on top, and it's been going through that resting period that we were talking about for about 5 hours. So what I'm gonna do now is just distribute these beads amongst all the cuvettes. Then I'm gonna cap the cuvettes and put it close to a window but outside of the heat, or just making sure heat's not penetrating the cuvette, and making sure that that CO2 indicator solution is cycling throughout the day and the evening. So purple during the day, yellow during the evening. And then once I see that the beads are doing that for about a day, then I'm gonna start setting up my experimental samples and then taking measurements every 5 minutes. So that will be the next part of this video. The easiest way to distribute these algae beads is you're gonna use 2 transfer pipettes. And so again, you're gonna open up the transfer pipette on the bulb side, and there is this skinny little portion on the transfer pipette. I'm gonna cut that off, and then I'm gonna cut up on the thicker portion of the stem at a diagonal to make myself a little scoop. Once I've done that, this is gonna serve as a scoop for my algae beads. And I will say that this part is a little bit tedious. This might be something, if you have a student that's looking for volunteer hours or something like that, they could do it for you, or it's something, even if you have a little extra time in class, which I know is unheard of, you could have the students do as well at their lab benches. So I'm just gonna open up this one as well, because this one's gonna be used to distribute the CO2 indicator solution throughout the cuvette. But all you're gonna do is open this up. I'm gonna, for consistency of a lab experiment setup, I wanna put an equal number of beads in all of the cuvette. The recommended amount is about 10, so I have a little scoop here. I already got 5 that I was able to easily scoop up, and I'm just going to kind of knock them off into the cuvette. If you have an issue, then the other transfer pipette that I have open can kind of be used to scrape them off the scoop. And right there, I think I have 10 in this cuvette. And so the next thing I'm gonna do is, the manual recommends putting 1 milliliter of CO2 indicator solution on top. I think I've done a little bit more in the past. I've done 2. It doesn't really matter. On this transfer pipette, 1 milliliter is pretty much the full length of the stem. So I'm just gonna take it out of this beaker and make sure that the algae beads are covered. That's really the biggest issue. I'm probably, from the looks of it, I'm gonna add 2 milliliters. And this cuvette is done, so I'm just gonna cap it. And what I'll end up doing is just laying this probably on this table that I have here with all my other cuvettes, and just moving it a little bit closer to the light to see that they're cycling through. The reason why I chose this many cuvettes, you don't have to do that. I actually am not sure if I even have enough beads to go through all of them. I think I do. The things that I want to test, I'm gonna do a cuvette that's pure light. I'm gonna do multiple abiotic factors at once. I'm gonna do a dark cuvette. I'm gonna do 3 that get filters with different quantities of light. So when I actually set that part up in probably tomorrow, I'll show you what filters I'm gonna use. I want to do 4 different wavelengths of light. So I have 4 cuvettes that I'm gonna wrap in cellophane. And then I have 2 that I'm gonna want to test the pH. One, they're gonna get algae beads that are soaked in vinegar, and one's gonna get algae beads that are soaked in Windex. And again, I think, like I told you before, this is one that I'm playing around with for this summer. I have tried the other samples in class with my students, but this is something that I want to try. All I'm going to do right now, like I said, is distribute out my algae beads, and then I'm going to push them over to the light and just make sure, at least for 24 hours, they're cycling in between turning the indicator solution purple during the day and yellow in the morning. And once I do that, I will finish up this video just showing you how I'm setting up all my different abiotic factors. So we're ready to start our experiment here. The first one I'm going to do for you is light intensity. So basically, I just want to show you the setup. I'm going to use 5 cuvettes for this one. I would take an initial reading of all of them, so they're all starting out about the same pH. So what I'm going to do for you in this video is take readings every 5 minutes. I will take a picture and kind of show you the pH. I'm sorry, this is a really weird angle. I have all this light kind of like shining off the cuvettes, but I will do my best to get a good picture for you so we can kind of estimate what the pH is of our samples as we go through this 45-minute time lapse. But if I were to look at it right here, I'd say that we're about a 7.7 to 7.9 pH. The material materials I'm going to use, I'm going to have one cuvette that's in pure light. One I am going to wrap in aluminum foil, so it's going to be in the dark, and I've made a sleeve here so we can get it on and off pretty quickly. The other thing that I have is I actually bought some filters that photographers use. They were really cheap on Amazon. That might be something that we have in our supply box later. The other thing that you can use is any sort of screening, or I'm not sure, maybe even something like wax paper or something like that. I'm going to basically just place these over my other cuvettes. The thing that I really like about these filters actually is that you can fit multiple student samples under each. If multiple groups in your classroom actually tried to do this, they could easily just slip their cuvettes under each of these, and that would save a lot of time in the classroom. So basically our initial reading, like I said, was about a 7.7 to 7.9. All my cuvettes are at that. So I am going to start my timer, and I'm going to turn on the light. I might move the cuvettes around a little bit just if there's a weird glare, but basically I'm just going to take everything off, snap a quick picture, and try to get the pH at every 5 minutes for 45 minutes. Okay, so the last experiment that I'm going to do today is just test different wavelengths of light. So all I'm going to do is take each cuvette and just wrap it here in this cellophane paper. I'm not going to count how many times I'm rolling it. I just cut them all the same length, thinking that I'll get about the same coverage. I'm sure you could make this more controlled and count it if you want. I'm going to do the exact same thing that I did with the light quantity and just unroll these every 5 minutes and take recordings. However, I'm not going to bore you with that. I'll just show you the end product, what I got in between all these different colors. So really, once the kids start getting into this and they just understand the idea behind the cuvettes, the world's really kind of their oyster. They can start testing whatever it is that they want with all these different abiotic factors. If I've got everything rolled up here, I'm still going to have a dark and a light as a control, and I'm going to turn on my light and I'll show you what I end up with at the end of this.